Yep.
Yes, now we can.
Okay. Okay. Hello, everyone. I'm Stefanos from the NTUA. Thank you, Mr. Vasilis, for the excellent presentation. I am working on the BioIndustry project in the WP5 package, which is responsible for building digital twins for 4 separate bioprocesses. And today I'm going to present to you some AI tools we have developed in the NTUA and how we imagine the digital twin for the bioprocess to be. So a bioreactor digital twin will consist of 2 different systems, your bioreactor system and also your cell. And these 2 systems are separate but also interconnected. And we visualize these models to be separate because we want to build 2 different digital twins, one for the bioprocess and one for the cell. The bioprocess digital twin will need reliable models in order to assess process performance and do online control to optimize the feeding strategies and the whole process. And the cell digital twin will be used in order to optimize the cell. As Mr. Vasilis mentioned, it's very crucial for metabolic engineering. It's very crucial for bioprocesses to use metabolic engineering in order to increase the strain performance. So how do we visualize the cell digital twin? Basically, it's as Mr. Vasilis mentioned, it can be a large-scale kinetic model. And large-scale kinetic model is nothing more than a set of ordinary differential equations for every reaction inside the cell's metabolism. And for every reaction, you need to find the kinetic parameters and also an intracellular metabolite concentrations. And to do this, and also if you do this, you can also simulate metabolite and flux changes and also test enzyme perturbations. And where can AI help with this process? First of all, Vasilis mentioned that they have developed tools in order for AI to increase the high throughput of stable and physiologically relevant kinetic models. But we are looking into a separate framework for this process in order to build a digital twin. It's not a different process, it's complementary. So generative models will be used to calculate kinetic parameters, and I will talk a little bit about generative models later on. Vasilis gave a really good introduction to Oracle. I'm not going to go into detail. I'm just going to pass through it a little bit faster. So first of all, when you're building large-scale kinetic models, you need to define the stoichiometry of your system. You also need to add thermodynamic information and also, if they're available, omics data, fluxomics or metabolomics. Then you can predict or sample thermodynamic ecosystem flux profiles and sample also metabolite concentration and thermodynamic displacements. This way you have created profiles of fluxes and metabolite concentrations, on top of which you can also sample kinetic parameters to create populations of large-scale kinetic models. These populations can be really large in number, and you need to prune them and do stability checks and any other pruning criteria you might have. And when you do this, you end up with some stable and physiologically relevant models for which you can do the metabolic control analysis, you can run dynamic simulations, bioreactor simulations, and also enzyme perturbations to design your next experiments. But let's say you have a population of such models and you do the next experiments and you get some results. What can you do in order to use the information from the kinetic models that you have to generate more models that agree with your new experimental data? And this is where our framework comes into play. Basically, using the population of generated kinetic models that you have, you analyze them on light of the new experimental data, and you train a generative model in order for it to sample kinetic parameters from a parameter space that produce these good results. And let's talk about how generative models work. Generative models try to learn the underlying data distribution so they can generate new realistic samples. These generative models are most often used in image processing, and some popular types are variational autoencoders, generative adversarial networks, and normalizing flows. Let's talk a little bit about variational autoencoders, because these are the models that we use at the time. And to understand the variational autoencoder, you need to understand how the autoencoder works. Basically, the autoencoder tries to predict X from X. Basically, it wants to compress the information from a data point into a latent space and then decompress it into the same values. This is really crucial for images, because if you have a high definition image and you want to transport it and send it to someone, you can compress it into a latent space, minimize the volume, and then decompress it using the decoder into the original form. But this is a deterministic algorithm. So a variational autoencoder is the probabilistic version of this AI model. It tries to represent the input data as distributions in the latent space. So if it is a distribution, you can sample from it. So thus making a generative model. So how we go about and building these kinetic models, we will show a case study that we are now running. For this use case, we have a small E. coli model with 90 reactions and 75 metabolites. It's the E. coli core model. And we have found in literature some gene knockouts and some fluxomics data, and from which we can calculate, approximate the flux control coefficients. And we have generated, using the Oracle framework, 12,000 kinetic models that are consistent with thermodynamics. They are stable, they are physiologically relevant. And for these kinetic models, we calculated the control coefficients and we compared it to the experimental ones that we have from literature. And for each model, we got a score, an error score, which was nothing else than the summation of the mean squared error of the experimental control coefficient with the ones that was predicted from the kinetic model. And we got a score for each kinetic model. And then we needed to create a data set. For this use case, we only used the saturation parameters, because these are the ones in the Oracle framework that are sampled through the elasticities. And basically, we split the data set into the top 30% performing models and the bottom 70%. And we did this because we wanted the autoencoder to learn not only to reproduce the kinetic parameters, but also to be able to classify the parameter space. To do this, we added another classification layer inside the variational autoencoder, thus making the training from unsupervised to semi-supervised. We wanted the autoencoder to learn the distribution and also to classify the good from the bad models. We did this. We trained the variational autoencoder. This is a PCA plot of the latent space. Sorry. As you can see, the parameter space is has very high variability. All the samples are really close to each other, and we were able to separate the two classes by adding the separation layer. And then we went into the generative. We went into the trained variational autoencoder and we sampled new kinetic parameters and we calculated new error scores, and we were able to lower the mean experimental error by 100 units. And also we got a new best model with a 150 score, whereas the first iteration, which was random, was 220. And this is really a really good indication that this model can work, and we are looking forward to running more experiments. And also we compare it with some experimental values, KM values from Brenda and Sabir K. and it has good agreement, but as Mr. Vasilis mentioned, they might be derived from different experimental conditions, so we do not look into them so much. And the key takeaways from this process is that AI can rapidly sample the parameter space and produce good quality large-scale kinetic models, and it can also improve predictions by integrating new experimental results. So it can work as a cell digital twin for your bioprocess. Having talked about the cell digital twin, we can now move on to the process twinning. For the process, we want to develop a new technology that is modular, generic, and capable to scale up. But the bioreactor system is really difficult to simulate because we have dynamics that evolve over time. We have different conditions, different states of the cell, induction, growth phases, stationary phases, and you can also have metabolic shifts inside your bioreactor, and all this stuff create uncertainty and difficult. Right now, we're talking about bioreactors, system of these for biomass, substrates, and products. It's really difficult because you might have non-linear and complex dynamics, you have linear noisy data, and also you have metabolic shifts, as I mentioned earlier. To this end, we propose a new framework that tries to do two things at once. First of all, you will have a machine learning model that is able to classify the best kinetic model for your bioprocess, and also another machine learning model that is able to do parameter estimation really fast. For example, if you have, for example, how this works, you have a model library with different types of kinetic expressions for your biomass, your growth, for your biomass production, your substrate uptake, your product secretion, and you do mix and matches of these models, and you create a kinetic model for your process. Then you generate in silico population of solutions, basically running a lot of simulation by sampling the parameter space for these kinetic models. And then you prune some solutions because you, for example, you do not want to have negative concentration inside the bioreactor. And then you use this data to train two different LSTM classifiers, which are deep learning machine learning models. And the first one is trained to classify your kinetic model, and the second one to do the parameter estimation. So when you have these models trained, you can feed it the experimental data that you produce in your lab, and it will tell you, okay, model number 10 works best for you, and I can also do the parameter estimation for you, and it will provide you with the parameters, and then you can have a good candidate set of these that works for your experiment. And STM models, long short-term memory models are deep learning models that are able to process sequential data, for our case, time series data. The good thing about these models in comparison to traditional recurrent neural nets is that they can manage long-term dependencies because they effectively capture both short-term patterns and also long-term behaviors. Okay, let's move on to a BioIndustry 4.0 use case, which is xylitol production from yeast. Basically, we have a fed batch bioprocess with two substrates. One of them is glucose, which is used for biomass growth, and the other one is xylose, which is converted into xylitol and is not converted into biomass at all. The metabolic pathway is cut off. And what we want for this bioprocess is to optimize the feeding strategy in order to maximize xylitol. And for this use case, we have 4 experimental runs, and let's see how we build the framework. This is the set of ODs, a general form of ODs that can explain this bioprocess, and this is the most complex one, where we have inhibitions from glucose, from xylose, from xylitol, and using this general form, we subtract terms and create permutations of this kinetic model. This matrix will show you which kind of kinetic parameters we alternated to create this family of models. And in total, we created 20 different kinetic models. And for each kinetic model, we generated 10,000 dynamic simulations by systematically sampling the parameter space. And this was the backbone for our LSTM training for the classifier and also for the regressor. For the regression, we trained different LSTMs for each kinetic model. For the classifier, we concatenated all the dynamic simulations and we trained it to classify which kinetic model was produced this time series. So this is a few words about the LSTM training. We had a 2-layer LSTM with heat dimensions 150, and the output was a softmax of 20 for 20 number of classes. The confusion matrix shows you how well the training went. We only had an issue with one class over here, but we had a validation accuracy of 95%, so we are quite happy with that. And how we trained the classifier, we went and did parameter inference into the experimental runs. We have the 4 ones that I mentioned earlier, and we have some additional ones which were with different conditions. So different conditions, I think, in different cells. So for the 4 top 1 experiments, the classifier was able to predict the same models most of the time. We had some ones that were we did not expect, but for example, we have model 12 and model 9 popping up most of the time. So we went back and looked into what common parameters they both had, and they had the xylitol inhibition in the product formation. And then to showcase how fast this framework can run, we went into model 12, which has cell death, inhibition for xylose and xylitol, and we saw that this is biologically relevant because xylose and xylitol share the same transporter, so it might be logical that they inhibit one another. So we went back, we trained the LSTM to do parameter estimation. Again, this time the regression loss was used for the training, and we used the data set of 10,000 simulations we have for model 12 in order for the LSTM to do parameter estimation. And then we went and did the parameter estimation for our experiments, and we were able to capture the dynamics of the system really, really well. And also this is for the experiment BC12, which all the simulations were based upon because we have fed batch conditions. And then we wanted to see how the other experiment runs performed using this set of parameters for this experiment, and also by running parameter estimation using the LSTM, because it will produce different parameters. And we saw that for both cases, we had some good results. We didn't predict biomass so well in here, but what we are happy with is that we had the kinetic expression that is able to capture the trends of all the species inside the bioreactor. And for some key takeaways for this methodology, because we're running short on time, I think, is that it can be used for offline applications in order for experimentalists to identify which model works best for them and do rapid parameter estimation without the need of optimization algorithms or any coding knowledge. And also, we are looking into how we can adapt this framework to work with real-time data, such as gas analysis, optical density, or Raman measurements, in order to try to predict metabolic shifts, because it's really important for the next framework that my colleague Kostas is going to showcase, which is the pink MPC framework for controlling the bioprocess. And this will be really crucial because you will be able to capture any metabolic shifts happening inside your bioreactor and act upon them. And with this, I am finishing my presentation and passing the torch on to Kostas.
Yes, we can.
Okay, I skip the presentation, the introductions, and I skip some of this introduced stuff that Stefanos also mentioned. Some incentives for digital twins and AI is that they can offer significant potential to improve and optimize bioprocesses, while some of the challenges are the limited volumes of data that we usually have available, and also the fact that these kind of processes are not easily reproducible as in other conventional engineering. So we are going to see some an open system demonstration of a semi-batch reactor. We'll see how we can develop, how we can generate digital threads from using the digital twin, and how the digital twin offers short and long-term predictive capabilities. And of course, how this kind of technology faces the challenges of limited data sets. The illustration case is the well-known experimental data set provided by INSA Toulouse. This process involves the production of recombinant proteins, specifically antivenoms for scorpion stings, and our purpose here is to apply twinning to a kinetic model in order to investigate the genetic instability of the modified microorganism. So the traditional regression-based or hybrid machine learning models require access to the full experimental trajectories. These data are only available once the process has finished, and we need all of this data in order to develop, to build a dynamic model in addition, this approach results in fixed parameter sets, which means that much of the connection to the real-time data is lost, limiting the ability to adapt or learn during the process. Instead, BioIndustry 4.0 employs a scaffold concept. This scaffold encapsulates all the available physical knowledge of the process and acts as a structured framework or matrix, if you will, on which the digital twin can be developed. In this way, we do not start from scratch, rather we build on existing mechanistic understanding while integrating new data. This is how the pipeline looks like. The key enabling technology for digital twin development is physics-informed neural networks. Unlike conventional neural nets that rely purely on data, PINs integrate known physical laws such as conservation equations directly into the learning process as soft constraints, making them data efficient, more robust, and better at generalizing to unseen conditions. As I said, we see the twinning process for a semi-batch bioreactor. We will start only with two data points, and we see that twinning is possible even with only these two data points. These data points are not the conventional data points used in standard regression studies, as they are complemented with the available process knowledge provided by the scaffold. Even without any prior knowledge, the twinning yields much better results, even at the point when it comes across the discontinuous changes in the state space. Okay, so we see how the predictions and the training goes. And finally, we see that we get a much better model, and the model which is aware of the full space. The inherited knowledge from the scaffold results in a deeper potential to predict discrete changes in the state space. Besides that, the twinning builds memory space that can be used in several ways. So we have two main remarks. First, we move from fixed parameters to digital threads that capture individual experiments. These threads may differ significantly to conventional representations, We can see that different process and experimental conditions could lead to distinct data threads. Digital twin provides process-aware and condition-dependent insights, and experiments could be linked to curated kinetics, connecting strain design to process engineering. In this point, we can somehow see and visualize how the work of Professor Vassili Hatzimanikatis and the large-scale kinetic modeling could be linked finally with our work and bridge the strain world, the microorganism world, with the process engineering. A second remark, of course, is PIN's ability to provide superior predictive capabilities compared to other traditional data-driven models. Even as the prediction horizon or rolling window increases, PINs outperform other data-driven methods, sometimes by 2 or 3 orders of magnitude. Okay, so we now move from model development to optimization and control, which of course remains some of the key challenges in bioprocesses. PID is the classic feedback controller. It measures the error between the set point and the actual process variable, and then adjusts the inputs accordingly. It's a simple and widely used, but it reacts only to what has already happened. MPC, model predictive control, on the other hand, is predictive. It uses a process model to anticipate the future behavior on the system and optimizes the control actions in advance. This predictive nature makes MPC very powerful and is already very popular in many industrial applications. The key question we want to address is how can digital twins enhance this further by providing more accurate, process-aware models for prediction and optimization. MPC works by predicting the future behavior of the process output and adjusting the inputs accordingly. Unlike PID, which reacts only to current errors, MPC uses a mathematical or a data-driven model of the process to forecast how the system will evolve in time. At each sampling time, MPC uses a model of the process to predict future trajectory of the control variable. The controller then determines a sequence of control actions over a prediction horizon that minimizes the difference between the predicted output and the reference trajectory. Only the first control action is implemented, and then the horizon shifts forward. The strength of this method lies in the ability to look ahead and optimize, but the accuracy of its predictions depends on the quality of the predictive model. That's exactly where digital twins can play a transformative role. The digital twin could build on this idea initially by replacing the simplistic linear models often used with the digital twin, or exploring more sophisticated solutions to optimization. A digital twin could also explore additional objectives, KPIs, as data are rich in context. Okay, this is how it looks like the prediction horizon shifts in time, and the digital twin builds memory and knowledge as new data come across. We perform some simulations, some in silico experiments for the control based on the data provided by Toulouse. This is the KPIs that we used. This is the standard KPIs used in control studies, but also we added a selectivity index, a KPI that measures the biomass formed per byproduct produced. The goal is to have a big value of this index, of this KPI. We perform several experiments with different noise levels and constraints. This is how an NPC pipeline could look like. And these are some of our results. This slide compares the performance of the control strategies under low noise and short reset times. With PID control, we see unstable behavior in the feed input and poor regulation of substrate concentration. ODE-based MPC MPC, which uses the ODEs, the discretized ODEs as model, improves stability, but still shows oscillations in the feed rate and deviations from the set point. Pink-based MPC, pink is a form of pins, but made with adjustments in order to be used in a control setting, delivers the smoothest and most accurate control. Biomass closely follows the set point, substrate concentration is well managed, and the feed input is stable. Here's a table of the results of this experiment. ODE-based MPC lowers the control effort, and in general, pink MPC outperforms ODE-based MPC across all defined KPIs. Here we see a similar experiment, but with higher levels of noise. Again, with PID, the system becomes unstable as substrate concentration rises to unacceptable levels and feed input becomes erratic. OD improves on PID, but still struggles with oscillation and constraint handling. Again, pink MPC outperforms other methods across all KPIs. Finally, even higher noise and longer reset times, which could be a more realistic scenario, we see again that the PID fails to hold the constraints. The ODMPC is better, but still have some struggle on constraint handling. And again, pink MPC seems to be better to outperform all other methods in all KPIs, especially in the selectivity index, which we have exceptionally high value of almost 815. Which are some conclusions that we can keep? It's that twinning is possible even with the sparse and limited data that we usually have in bioprocesses. The superior predictive performance of pins of these process-informed neural networks, which surpass the other data-driven methods, linear and neural nets, by sometimes 2 or 3 orders of magnitude. And maybe we want to highlight this: the digital threads that we generate serve as footprints of individual experiments. This could be a potential integration with metabolomics and genome scale metabolic models. This will finally build a bridge between these two worlds. In addition, we see that the pink offers some advanced control capabilities, as it was able to outperform conventional controllers in almost all the KPIs that we defined.